Hello everyone. I hope you all are doing great. Welcome to my YouTube channel, Micrologue. The channel Micrologue is truly dedicated to the students who are dealing with microbiology subject in their syllabus, and the notes will be helpful for the microbio students, pharma and nursing students as well. Apart from the basic microbiology, food microbiology will be discussed in later series. So without wasting much time, let's start with our first video, that is Culture and Identification of Bacteria Part 1, lecture by Microlog. As we already know that microorganisms, they exist as a mixed population in any ecological niche or in any residential flora. In order to identify such important microorganisms, we must need to isolate them from the samples in their pure form, and this can only be achieved by growing the samples in a specified medium. This slide representing the basic requirements that are needed for the medium to grow bacteria or fungus. The most common ingredient is peptone with complex mixtures of digested proteins including proteases, polypeptides and amino acids as well. Apart from the peptone, there are some inorganic salts as well, such as potassium, phosphates and magnesium. Some bacteria require special growth factors for their growth, such as riboflavin, and other microorganisms may need blood, serum, and yeast extracts for their growth. Now, the classification. Bacterial culture media are classified on the basis of their consistency, constituents, and their functional requirements. Based on the consistency, we have three types of media, that is liquid media, solid media and semi-solid media, which is also known as sloppy agar and is most widely used in microbiological analysis. First one is liquid media. This media is mostly used when we need large volume of samples, and they are used for the preparation of bulk cultures like in antigens, in vaccines, and in secondary metabolite productions. Second is the solid media. These medias are mostly used for the isolation of bacteria to have distinct colony morphology, to study their pigmentation and hemolysis patterns like alpha, beta, and gamma hemolysis. The basic of any medium is agar, and it is obtained from a type of seaweed. I have also added a sticky note over the presentation, that is agar agar is mostly obtained from algae, which belongs to the genus Glacilaria, Gellidium and Eushioma. Agar usually do not have any nutritive value, but also it does not affect by the growth of bacteria. The melting temperature of the agar is 98 degrees Celsius, while it solidifies best at 42 degrees Celsius. Third one is the semi-solid media. Semi-solid media usually have low concentration of agar, that is from 0.5 to 1%, but in some cases we have seen that the concentration may vary from 1 to 15% as well, and it is used to demonstrate the motility of bacteria. Classification based on the constituents. So on the basis of their constituents, we have first one is the simple media, which is also known as the basal media. Nutrient broth and nutrient agar are the best examples of basal media. In case of nutrient broth, it consists of peptone, meat extract, sodium chloride and water, and it is used to demonstrate the motility in bacterial growth. Second is the nutrient agar. It is made by adding 2% agar to the nutrient broth, and it is the simplest and most common solidified medium used as a basal media in clinical diagnostic laboratories. And it is also used for the demonstration of pigment producing ability of bacteria. Second one is the complex media. These medias have added ingredients for the special purposes, such as the bringing out of certain characteristics of bacteria or providing the special nutrients for the growth of bacterium under study. Third one is the synthetic media. These media are usually prepared from the pure chemical substances, and the exact composition of the medium is fully documented. They are used for the various special studies, such as their metabolic requirements needed by the bacteria. Third one is the classification based on their functional requirements. So first one, we have the enriched media. uh, Enriched media have some nutritional requirements such as blood, serum, eggs. They are added to the basal medium in order to cultivate the fastidious bacteria. Examples of such bacteria are blood agar, chocolate agar, and brain heart infusion agar. The blood agar is a solid culture medium with the agar peptone and blood in it. And the sheep blood is usually used, but we can also use horse, cow, and pig blood as well. It supports the growth of most aerobic and anaerobic bacteria, as it have special components in it like vitamin K, cysteine, hamine supplementation, which promotes the growth of anaerobic bacteria and some fungi as well. Blood agar is also used as an indicator agar, as it indicates the degree of hemolysis caused by hemolysin. And on the basis of this, it is used to differentiate among the gram-positive cocci, and it can also known as differential medium. Next is the chocolate agar. The chocolate agar is a mixture of sheep blood and nutrient agar media, and this results in the release of hemoglobin, a related substance called hamine, and also known as X factor, and nicotinamide adenine dinucleotide NAD. And during the process of heating, it is used to grow mostly the fastidious organisms like Haemophilus influenza, and meningitis, and gonorrhea, and pneumococcus species. Third one is the brain heart infusion broth medium. It is highly nutritious medium, or we can say buffered fluid culture medium, which is prepared by non-enzymatic infusion. Brain heart infusion is prepared by the non-enzymatic infusion from calf brain and cow heart, and added with peptone and dextrose medium, and it is best suitable for the cultivation of fastidious organisms. Second one is the enrichment media. These media are used to suppress the commensal bacteria while allowing the pathogen to remain viable and to grow. And some inhibitory substances are also added to the medium, like some of the examples of the enrichment medium are tetrathionate broth and selenite F broth. I repeat, tetrathionate broth and selenite broth. Tetrathionate broth inhibits the growth of coliforms, while at the same time allowing typhoid paratyphoid bacilli to grow. On the other hand, selenite F is used for the fecal samples in case of dysentery. Third one is the selective medium. Okay, as in this case, the selective medium have addition of an inhibiting substance to the desired medium, which allows the growth of required bacteria while inhibiting other bacteria. Example of such media are deoxycholate citrate agar media, which is used for the isolation of bacteria from fecal samples. Next is the thiosulfate citrate bile sucrose agar media, which is used for the isolation of Vibrio species in case of cholera. And alkaline pH supports the growth of Vibrio. That's why thiosulfate citrate bile sucrose agar is considered as best for the isolation of Vibrio species. Apart from deoxycholate agar and TCBS medium, there are two other mediums which is known as Thayer-Martin medium and Lowenstein-Jensen medium. I repeat, Thayer-Martin medium and Lowenstein-Jensen medium. Thayer-Martin medium is used to isolate gonorrhea, Neisseria gonorrhea from urethral and endocervical swabs, and LJ medium is used for the isolation of Mycobacterium species. Fourth one is the indicator medium. Okay, these medias contain an indicator which changes color as the bacteria grows on it. The best example is Wilson-Blair medium and McLeod's medium. As in case of Wilson-Blair's medium, there is a presence of a compound which is known as sulfite, and when Salmonella typhi grows on Wilson-Blair medium, as typhi reduces sulfite to sulfide to give a black metallic sheen on the colony. On the other hand, potassium tellurite in McLeod's medium is reduced to metallic tellurium. I repeat, it is reduced to metallic tellurium by Corynebacterium diphtheria and produces black colonies. Next is the differential media. So we have a list of differential medias including McConkey medium, which differentiates lactose fermenters as pink colonies, and the non-lactose fermenters appears as colorless or pale colonies. Another example is cysteine lactose electrolyte deficient medium, CLED medium, which is used for the isolation and enumeration of bacteria from urine sample. Next is the transport media. These are the special media which are used to transport specimens suspected to have fastidious organisms. Okay, which microorganisms? Fastidious microorganisms. Example of such transport media includes Stewart's transport medium and Gary-Blair medium. Stewart's transport medium is used to transport specimens which have gonococci bacteria in them, and Gary-Blair medium is used for the transportation of enteric pathogens. Next is the anaerobic media. These are used to cultivate the anaerobic microorganisms like Clostridia, bacteroids, and they may contain some reducing agents as well. The best example of anaerobic medium are Robertson cooked medium and thioglycolate medium. And as in case of Robertson cooked meat medium, because there is a presence of meat particles and dextrose, yeast extract, hamine and vitamin K, this medium supports the growth of anaerobic microorganisms very well. Next is the thioglycolate medium. As this medium contains some special ingredients including yeast extract, calcitone, sodium chloride, L-cysteine, thioglycolic acid, agar, methylene blue. So it facilitates the growth of anaerobic, aerobic and facultative anaerobic microorganisms very well. Next is the defined or synthetic medium. These are the media whose constituents are clearly defined in terms of each ingredients. Their constituents are clear. They are chemically defined media and they are prepared from the pure chemical substances. Example is minimal essential medium, which is mostly used in tissue culture labs. So this is for the today's lecture. I hope you like the video. If you found the content relatable, please like, share and subscribe to the channel. Thank you so much.